My name is Mrinmayi Kulkarni. In this poster, I'm presenting some data from a project examining the neural correlates of category specificity in associative memory. An important aspect of episodic memory is the ability to flexibly recombine distinct elements across episodes to form new associations. For instance, let's say that I see a woman with a dog in the park, and at a later time I spot the same dog with another person. I might infer that the man and the woman either live together or jointly own the dog, even though I've never encountered the man and the woman together in the same episode. Past studies have shown that a network of regions, including the prefrontal cortex and the medial temporal lobe regions, support such associative inference in memory. We also know that the medial temporal lobe structures exhibit category specificity. For instance, studies show preferential processing of scenes in the anteromedial hippocampus and the parahippocampal cortex. On the other hand, the fusiform face area and the ventral anterior temporal lobe, including the perirhinal cortex, are involved in face processing. So in the current study, we aim to examine whether MTL subregions also differentially support category-specific associative inference in memory. To test this, we recruited 31 participants who completed 8 runs of the associative inference task while undergoing fMRI scanning. In the study phase, they encoded overlapping face-object or scene-object pairs. For instance, in the beginning of the run, they might be presented with this red chair paired with the playground scene. Later in the study phase, they see the same playground scene paired with a spoon. In the corresponding test phase, they're tested on their memory for the direct associations. So for instance, they might be presented with this face as a cue and they have to pick the object that was associated with it. They're also tested on their memory for the inferred indirect associations. So here they might be presented with the chair and they have to pick the spoon that was never directly paired with the chair but was linked via that common playground scene. Across runs, the A and the C items were pictures of everyday objects, whereas the B item could either be a face or a scene. Here are the behavioral results from the task. We have accuracy on the y-axis in the direct and the indirect test phase, with scenes in red and faces in the gray bars. We found a main effective test type with worse performance on the indirect test as expected, but there was no difference in performance between faces and scenes. With the fMRI data, we first looked at whether there were any brain regions that predicted subsequent memory for indirect associations. To do this, we backsorted study phase trials as a function of whether the corresponding indirect test trials were subsequently remembered or forgotten. Then we contrasted the correct versus incorrect trials separately for scenes on the left and faces on the right. Here we found that the hippocampal activity predicted subsequent memory for indirect associations for both scenes as well as faces, but there was a hemispheric difference such that activity in the right anterior hippocampus was higher in correct trials involving scenes, whereas activity in the left anterior hippocampus was higher for correct trials involving faces. Next, we looked at activity during the indirect test trials in sensory regions in the temporal lobes. We first identified regions that might be involved in the sensory processing of scenes and faces by contrasting direct test trials involving scenes and faces. As expected, here we found higher activity in the parahippocampal cortex for scenes and in the fusiform face area for faces. We then extracted activity from these clusters from the indirect test trials. In this graph, I'm plotting percent signal change on the y-axis in the indirect test trials as a function of the category of the linking items. The top panel, we have activity from the parahippocampal cortex, and in the bottom panel, we have activity from the fusiform face area. The green bars are trials that were correctly identified indirect associations, and the pink bars are the incorrect trials. Here we see that in the parahippocampal cortex, activity was greater in the indirect test trials linked by a scene as compared to trials linked by a face, even though no scene was actually presented on the screen in these trials. We did not find a similar effect in the FFA. There was no difference between categories in this region. Finally, to examine the involvement of different MTL subregions in category-specific associative inference, we manually segmented MTL subregions on each participant's high-resolution T2 image as shown here. We then extracted activity from the indirect test trials from these regions in the left and the right hemisphere. I'll walk you through the significant results. We first found a main effective category in the left and right posteromedial entorhinal cortex with higher activity for scenes than faces. We found a main effective accuracy in the bilateral hippocampal tail and the right CA1 with higher activity for correct trials as compared to incorrect trials. And finally, we found a category by accuracy interaction in the bilateral CA2-3 and dentate gyrus, the left CA1 and the right perirhinal cortex with higher activity for correct than incorrect trials but only for scenes. To conclude, the goal of the current study was to examine category specificity of the MTL subregions in associative inference in memory. Here we found that hippocampal activity during the study phase was predictive of successful associative inference, but there was a hemispheric difference in the hippocampal involvement for scenes and faces. We found that activity in the temporal lobe regions involved in the sensory processing of scenes, so the parahippocampal cortex, persisted during tests of indirect associations involving scenes, even though no scenes were actually presented on the screen in these trials. Finally, we found that involvement of some MTL subregions in tests of indirect association depended on the category of the linking item and the success of the associative inference. Consistent with past work, our results highlight the specialization of the anteromedial hippocampus in associative inference involving scenes. And we think that associative inference involving scenes may recruit the hippocampus to a greater extent than faces to aid in spatial relational binding that is specific to scenes. Thank you.